Hello, my name is Anna Toruńska-Sitasz. This short video covers the topic of sampling in the marine environment. Collecting organisms is the first stage of the path from organism to product. Nature has always been a source of inspiration for design and technologies, as well as for biotechnology. Currently, natural products are mostly developed from compounds produced by terrestrial organisms. However, due to enormous and unique biodiversity of marine habitats, a shift to marine sources has become clear in recent years. So-called marine bioprospecting can be described as exploration of marine biodiversity for new biological resources of social and economic value. This may include all kinds of organisms and areas of application, including medicine, food, feed, and biofuels. Challenger expedition was the first one organized specifically to gather oceanographic data and to explore the diversity of marine life. During this expedition, hundreds of new genera and nearly 5,000 new marine species had been discovered from the surface down to the seafloor. From that time, many tools were created and methods were developed to sample marine waters and different kinds of sediments. We also know how to sample aquatic organisms belonging to neuston, plankton, nekton, or benthos. But does it mean that the ocean is well sampled and we have a comprehensive knowledge about the marine life diversity? The first reason why the answer to the previous question is no is that most of the marine biosphere is very difficult to access. It's not an exaggeration to say that we know more about the surface of the moon than how the bottom of the oceans looks like. Only about 10% of the seafloor has been mapped with ship's sonar, producing images with resolution up to ±100 meters in the deepest areas of the ocean. And even if we can reach the seafloor, the only way to sample organisms at such depths is with a remotely operated underwater vehicle. The tools used by ROVs are designed to operate at high water pressure around them, but they can end up crushing the fragile marine organisms before the scientists even have the chance to study them. Soft grippers are one of the options for better organism sampling. Soft robotics are cutting-edge technology. Moreover, during the expedition into the depths, organism-adjusted manipulators that collect the fragile specimens can be 3D printed on board. In this example, soft robotics manipulators were tested down to over 2,000 meters via a remotely operated vehicle in the Phoenix Islands protected area and facilitated the study of soft-bodied life. Another problem that we face while sampling the ocean with traditional equipment is loss of biodiversity of the smallest, but still the most numerous organisms, the microbes. Why? Because most of them are uncultivable. The help to study the uncultured comes from metagenomics. Metagenomics represent a new approach in genomic analysis. This method is based on the culture-independent cloning and analysis of microbial DNA extracted directly from an environmental sample. Metagenomics facilitated the discovery of many novel genes, metabolic pathways, and even whole genomes of uncultivable microorganisms from various habitats. Still, the limitation of this method would be sampling extreme locations in order to collect extremophiles. A good example of metagenomics' usefulness to study life in the ocean is the 21st century Sorcerer II excursion. During more than 2 years, Sorcerer II circumnavigated the globe and collected one of the largest metagenomic datasets. Millions of new genes, new lineages of viruses, bacteria and eukaryotes, and nearly 1,000 genomes of uncultivable lineages of microbes were annotated. Sampling in the marine environment is an important part of bioprospecting, especially in case of biodiversity studies. However, due to rare occurrence and protected status of many valuable marine species, collection in the marine environment is not an option when our bioprospecting goals are biomass or compounds. An alternative to collecting organisms would be culturing them. The other ways to obtain products based on marine organisms' metabolites are bioengineering and synthetic chemistry. Thank you.